Good evening, everyone. I'm Yunhai Wang from Shandong University, China. Thanks for taking your time to join today's CSIG-VIS International Lecture Series. It's my great pleasure to introduce our today's speaker, Viola. Viola is a professor at King Abdullah University of Science and Technology, KAUST, Saudi Arabia. Graduated from TU Wien, Austria, and in 2005, he took a postdoc position at the University of Bergen, Norway, where he was gradually promoted to professor rank. In 2013, he received a WWTF grant to establish a research group at TU Wien. At KAUST, he continues developing new technology that make visual in silico life at the nanoscale possible. Moreover, Viola co-founded the nanographics startup to commercialize nano-visualization technology. Viola and his team in nanographics create the 3D structure of coronavirus that has been shown in all CCTV in last January. If I remember correctly, I watched that. The video showed that. Yeah, that's very impressive. Today, he will talk about his work in volume visualization for Cryo-Electron tomography. Okay, before we start, we encourage everybody to join the WeChat group by scanning the QR code here. And in the WeChat group, please feel free to drop questions about today's topic. And you can also raise your question here in Zoom. We'll answer your question at the end of the presentation. Okay, now let's welcome Professor Viola. Yeah. Ivan, please take turn.
Thank you very much, Yunhai, for your kind introduction. Now, if you don't mind, I will share my screen as well.
Yeah, please.
I will then start my presentation. Today, I would like to talk about the topic which we have recently started at KAUST, and it was the trigger was a collaboration with Tsinghua University, who actually have been working on the tomographic acquisition of the SARS-CoV-2 specimen. And based on their data, we have sort of realized that we need to engage in some new research in volume visualization, with particular focus on the cryo-electron tomography, Cryo-ET. But before that, we have been working over several years on visualization of atomistic models, which were not generated by the microscopy directly, but they have been created as a result of multiple research teams working on uncovering some structural details of particular biological structure, like such as viruses or bacteria. You can see some of our work on our website, nanovis.org or nanovis.kaust.edu.sa. I'm welcoming you to check out the publications, but also the resources which are there, which might be quite interesting for you to use as models or code or software. All this is available. Before we started to focus on the cryo-electron tomography, we have worked on visualization and rendering topics of these what we call mesoscale models, which are models of 100 millions of atoms, and they form ultrastructure of viruses and of their surroundings. So under ultrastructure, I mean that we see the macromolecular composition and the envelope, the biolipid membrane, and the genetic macromolecules. So the other small molecules and water are not present in our models. And later on, we realized that not only we should be focusing on visualizing and rendering these type of models, but to actually develop a technology to to create models quickly in a form of a visual programming environment. So the idea was that that there are rules how the ultrastructure is composed, for example, of a virus. The spike proteins on the envelope here, for example, are placed according to some rule. Their distribution is based on some observations and so on. And all this can be specified in our modeling system. And this specification is then a basis for packing or for creating these ultrastructures. So we have rules, and with these rules we can generate many models which are statistically identical, but structurally they are, of course, always a little bit different from each other. So now we have sort of connected our previous work on visualization with also some technology for authoring. This has resulted in a tool called Mesocraft, and it's, for example, downloadable from our nanovis.org resources website. Currently, this tool is used with domain experts in order to model various interesting parts of cellular mesoscale. But once we have released the model, which we have generated at KAUST, which you can see here, and it's also stored at the KAUST repository, we have been contacted by Sai Li, a scientist and professor at Tsinghua University, who wanted to revise the model of the SARS-CoV-2 ultrastructure with us, because the original version which we had was somehow differing in the area of the internal part of the virus. So we have started to collaborate with Sai Li, and that was also the basis for generation of various videos of this virus, which you have seen both in terms of atomistic model, but also of the volume rendering actually of the specimen, which Sai Li has imaged and then kindly provided to us. You can see how such a specimen looks like after tomographic reconstruction, and also after several image processing operations. You can see that clearly some structure, some specimen is recognizable there. If you don't mind, I move this on this other side. So, and however, you can still see that there is a lot of noise, and this noise is somehow occluding the interesting structure. In 2D, we can see the specimen. You can imagine that in 3D, all these little speckles which are here will be occluding the specimen, which is kind of centered in the middle of the volumetric tomographic reconstruction. So, but already we sort of realized that maybe now is the time where the resolution of the microscopy is so good that we can actually generate atomistic models or relate atomistic models with the microscopic experiments or with the microscopic acquisitions, and started to look more deeper on what technological standpoint do we have in terms of visualization of cryo-electron tomography data. And a lot can be adapted or adopted from traditional volume visualization, but it has to be, adjusted for this particular case of especially strongly high presence of noise. So I will maybe just list what are somehow the new challenges with in volume visualization of cryo-ET. It has a very low signal to noise ratio, and that is for a reason, because if we are doing the so-called tilt series, which are the basis for the tomographic reconstruction, we need to limit the dosage of the specimen of the electrons. Otherwise, the specimen will degrade, and we will be not having the original untouched specimen anymore to image. So this is somehow a limitation which then results into low signal-to-noise ratio. If we would need only to image one projection and not several in the tilt series, then we can use much higher voltage. But for creating a volume, we will need to restrict ourselves to low voltage, and that implies the low signal-to-noise ratio. And then on top of that, we cannot image the tilt series from all possible angles, because the specimen, how it is placed, only allows us to the tilting by of the specimen by plus minus 60 degrees. So, which means that we have still some area from which or angles from which we are not imaging, which we are not able to image our specimen. So, and this results into missing wedge artifacts, and I can point out to those at some later point in time. So, but not only challenges are coming with this modality, but also opportunities. The opportunities are not only about the cryo-electron tomography modality, but about the nature of imaging the biological specimen. Here we have, for example, in mind that often if you are imaging something with cryo-electron tomography, you will have often the same structure present, or a very similar structure present in the data several times. So, and this can be used, and in fact it is used for techniques like subtomogram averaging, where you are picking these various elements from the volume, you are co-registering them so that they overlap, and through the approach of superimposition, you can combat noise, because the signal will be the same after the registration, or very similar among these individual instances, while the noise will vary. And based on the superimposition, you can then cancel out the noise by having several samples superimposed. And also, if we, for example, learn how to interpret some structure in the data, this interpretation can be then reused, can be used multiple times, and not only just for one particular object within the volume, because we have so many instances. And also, this is something which we have been experimenting with, and I will give you a glance into that. If we know the imaging process with the current trends of differentiable approaches and neural rendering, we can actually think of reversing the imaging process and trying to then recreate the specimen out of the acquisitions, out of the projections. But more about that just in a moment. Let's first talk about our work on tomographic reconstruction, or what is done in tomographic reconstruction in general, and then in particular in our case. You can see here, hopefully, I hope, you can see here the specimen which has been provided by Sile's team to us, which are the tilt series of the intact SARS-CoV-2 variant. What you can see here are several things. So you can see these dots, they are most prominent. And these are in fact not very interesting, because those are just some gold bead particles which are useful for aligning the tilt series. So those come typically misaligned, and you would like to align them, and these beads serve the purpose of alignment. Then you can see some other structures on the border where some other biological specimen is present. And of course, you can see here as well the SARS-CoV-2 variants, several of them. And now we have the tilt series, plus minus 60 degrees, and our task would be to generate a volume, so a regular grid volume with intensity values, so that we can then perform the volume rendering. So, and this is I will now describe the Beer's law. The Beer's law, which is used in the tomographic reconstruction, but which is perhaps a simplification of the case for electron microscopy. But it will serve us the purpose of understanding how the attenuation of our signal through the transmission electron microscopy process is done. So imagine that we have now some medium and we are passing through it some energy. It could be light or the electron beam. And so this is a ray. And before we enter the specimen, we have some intensity 0. And at the end, we are attenuating because we have some kind of density here. We are attenuating this intensity, and this intensity is after passing through this specimen is attenuated to the intensity R. So this is also R because it's for this particular ray. And well, the density can be homogeneous, but also it could be varying. So this is a profile of the density along this ray R. Now we can use the Pierce law principle in order to learn how does what law does this attenuation follow, and we can see that there is an exponential fall off of the intensity, which is of course dependent on the changes in the density, which is expressed in the exponent. Now this is a little bit unfortunate formulation. We would like to have a simpler formulation, and for that in tomographic reconstruction, we would often do some changes to this equation and put, for example, the original intensity to the left-hand side of the equation, and then to get rid of the exponential function, we perform logarithm of this equation. So at the end, we are ending up with something which is a sum of densities along the ray. So in a discrete case, this will not be an integral, but it will be a sum. So if we have in our case, we would have some kind of a specimen, which we do not know at the beginning, because what we know of this coming from the acquisition process, we know the projections. And now imagine that I'm drawing it from the in 2D or from the top view somehow. So we have several projections. Ideally, we have from several directions, but sometimes this is not possible, like in the case of the microscopy, electron microscopy. So, and here we know the projections, and now I will call this this type of projection. So after the normalization and the logarithm, I will call it a P. So, and these are the projections P. And now we don't know what are the values inside, but we know that these values can be somehow have contributed by means of a sum along a ray. So, and this, as we have a lot of projections, we can use various iterative optimization techniques or non-iterative techniques as well in order to perform the reconstruction of the volume. So, and how is this done? So if we have our projection, or maybe I draw it a little bit aside. If we have our projection, then for one value on the projection plane, imagine that we have our volume, which consists of discrete values, which are somehow equidistantly present. And now this ray is sampled at some some distances. And actually, these are reconstructed values from the samples in the discrete representation of the volume. Now, what we can do at the beginning in so-called algebraic reconstruction techniques is that we initialize our volume with some value. For example, we could use an average value, so gray level value everywhere. And then what we do is we perform a so-called forward pass. So for our array, forward projection. And with this forward projection, we can generate a new projection. So this is the ground truth. And we will create something like the first iteration of the projection. The first iteration will be just a sum through a homogeneous medium if in case we are initiating it with maybe just the average grayscale value. But then we will be subtracting the first iteration from the ground truth, and we will generate something which we called an error, which will express how far we are with our through our forward projection from the ground truth. And then what we can do with this information, so we can take the error values, and so we have our volume again, and these error values as our array was passing through these samples, so this volume should be the same. As our array was passing through these samples, we distribute the error to all of the contributing samples through a back projection. So we back project the error into the volume, and with this we are actually improving the estimate of how this volume looks like. Now, we don't do this only for one ray, but we do it for all the pixels, if you like, on the projection plane. And we calculate the error term for each of these. And not only for this one direction, but we can do it for all the available directions. And we can do it in several iterations over and over again with trying to reach a state where the error term is getting smaller and smaller until it is so small that we are satisfied with our reconstruction of the volume. And that leads us to the reconstructed volume as the regular volume, which can be used for volume rendering. Now, in the case of our Cryo-ET, when we have our projections, so we have here our volume, let's say, and we have one projection, and we have another projection and another projection as the border ones. So this would be 0, and this would be let's say, or yeah, these two will be plus minus 60 degrees. So we have and we have all other projections in between, of course, between these field series, but we do not have more projections beyond the 60 degrees, which means that in spatial domain, these are projections, but with the projection slice theorem, we know that in the frequency domain, this corresponds to a slices. So the projections correspond after transformed by Fourier transform, they would correspond to slices through the volume with the same angle. So we will have slices which look like this, but maybe a little bit more. But So here we will have a frequency information about the volume from this acquisition process in these areas, but here we have still unknown. So these frequencies are missing, and that's why it is called a missing wedge. It's a missing wedge in the frequency domain. So this will affect the quality of our reconstruction. After we perform our reconstruction as described with algebraic reconstruction techniques, we can see that the tilt series are changed into individual slices. And we have created successfully a volume, but this volume is still quite noisy. And normally, either the projections are denoised and then the tomographic reconstruction is done, or the tomographic reconstruction is performed and then the volume is denoised somehow with various approaches, from standard denoising techniques up to some deep learning based techniques. But in our case, we have realized that in tomographic reconstruction of cryo-ET data, we have the tilt series information. So we have the original projections, which is quite different from the situation with computed tomography, where typically the scanner will already perform the tomographic reconstruction and the access to the projections is original projections is not available. So here we actually know the ground truth of the projections, and we can, during the tomographic reconstruction, we can check always how far we are from the ground truth of these projections. And also, we can, in fact, inject the process of denoising in the tomographic reconstruction. And by denoising, after the denoising stage, we can always check whether we went too far away with our denoising and we have increased the error in undesirably, or whether we have actually favorably improve the quality of the reconstructed volume by the denoiser. So the bottom line is we want to make the denoising part of the tomographic reconstruction process such that we can always, by checking the error term, control the level of smoothing, regularization, if you like, of the denoising process. So we are having several approaches of denoising. There could be various ways. One commonly used would be the total variation, which is a total variation is something like if you have a signal, then you have a very noisy signal, but you would like to achieve that your denoised signal is somehow close to the original signal. And at the same time, so this would be maybe the denoised value. At the same time, it is not so far away from the neighboring denoised value. So here, sort of the two aspects close to the original values and close to the denoised values is driving the iterative process of denoising using total variation. So the result of the total variation will then look finally approximately like this. And this we can, the total variation, we can then include into the process of the tomographic reconstruction. So imagine that if we have our projection, and this is the, let's say, the ground truth, and then we have our volume, and well, we are calculating our ith iteration. Then let's say for this ray, so we have here some of the values, and we are summing up, we are doing the so-called forward projection pass, and then we generate the ith iteration of our projection. Now, before we perform the comparison or the relation through in order to generate the error projection, we can perform the denoising on this projection. So then after the forward projection pass, we do the total variation on the slice, on the projection. And afterwards, we subtract these two values and create the error term projection. So this is, in a nutshell, the reconstruction process. And here we can, of course, with several parameters, control the amount of the contribution from the regularizer or from the data term. So which means that of the simple comparison between the ground truth and the ith iteration. And compared to when we include the total variation or other denoiser, we can see how does this impact the final reconstruction of our volume. Please note that as it is part of the tomographic reconstruction process, we always have control over how far we go from the original projection. We do not have this information available if we perform the smoothing on the already reconstructed volume, or if we do it uncontrollable on the projection first. So you can see here another volume of I think some HIV particles or envelopes of the HIV, and on this in this volume you can clearly see on the right hand side that the denoiser has contributed in the tomographic reconstruction to much clearer depiction of our of the specimen than what you see on the left hand side. What you can see are these black dots. These are simply the fiducials, these gold beads, which have been used for the alignment of this tilt series. We can also see the benefit for 3D volume visualization. So if we have the noisy reconstructed volumes, and we are trying to use our direct volume rendering techniques, we see that the visualization is dominated by the presence of noise. If we use the denoiser, and in our case in a controlled way, we can see that the specimen clearly stands out, and the noise or the background variations are significantly suppressed. So this is somehow a classical way how we would approach the tomographic reconstruction. And now that we have access to the ground truth, we can already check even during the denoising process how far we are from the ground truth and what type of errors are we introducing. Quite a different way to look at it would be to take the recent trends from AI, machine learning, and also computer graphics to try to mimic the microscopy process and evaluate which parts of the microscopy simulation we can actually realize as the differentiable ones. And if we can make them differentiable, this means that we can solve so go backwards in the microscopy process, meaning going from the finally rendered tilt series to the representation of our specimen. So we can, the traditional way of simulation will be this way. So we have at the beginning some atomistic representation of our virus. In this case, this is Zika. And this is one intermediate representation along the microscopy simulation or transmission electron microscopy simulation pipeline. And after adding noise, we can see that this is how the projections will approximately look like. So, but with if this pipeline is made differentiable through optimization approaches, we can hopefully go backwards. And we have started to look into this and have some early results to report on and to share with you. So the electron microscope is in some sense analogous to an optical microscope, which we can often relate to much easier. So it has analogous components, but they are realized in a different way. So we have the source is an electron gun instead of a light source, and then or an X-ray source, for example, and then we have the lens, but the lens are basically coils which influence the path of the electron beam, and are able to defocus it or focus it according to the voltage which is in these individual elements and on their other physical properties. So maybe it's not completely important to describe all of those, but we can see that there are several lens systems, and there is in the between, there is a specimen located in this area, and this electron beam eventually, in magnified form, interacts with the detector. So the detector will then transform the electron energy into a light energy, and will allow us to see this magnified specimen in the detector. And this detector has several properties. And so far, we have been looking at how can we, by means of differentiable approaches, how can we change, or how can we reverse the process of processes which are going on in the detector. So here on the left-hand side, you can see that our transmission electron microscopy simulator consists of a part which is dealing with the propagation, wave propagation. It involves the coils, the magnifying lens are acting actually as a Fourier transform, and Fourier transform is, for example, differentiable. So these parts are to a large degree differentiable. We are still working on it to make it differentiable. We are not complete in this part, but we have so far focused on the differentiability in on the detector. So, and here, an intermediate representation coming from this wave propagation process, an interaction of the electron beam or electron wave with the specimen, is a representation which looks like this. It is not containing a noise yet, and it is a basis for the noise generation, because the specimen itself influences the noise. The noise is not independent from the specimen. And in fact, with the CMOS detectors, we can model the noise as a shot noise with this, which is a Poisson process. So this term quantization here is adding the noise onto the signal. And then in the detector, we have further parameters, something which is called the MTF function, which is in the frequency domain, or its spatial domain counterpart, the point spread function. So, and these are functions which are basically defining us how blurred is the signal on the detector. So the point spread function will be the convolution kernel which can be used for the blurring the signal. And this MTF, so it's a frequency domain counterpart, because this process which we have here is carried out in the frequency domain, can be characterized by several parameters. So this MTF function will typically look something like this. It will be somehow a curve, symmetric one, and the point spread function will be the its spatial domain counterpart, and that will look approximately like this. So small blurring kernel, and we can characterize this MTF by these parameters which you can see here. All the blue parts are basically defining you on this schematic diagram of the differentiable simulator. What are the parameters? And these parameters are used for representing the MTF. And by the differentiable approach, we can actually by giving the system some representative projections, we can learn this, or we can by means of optimization estimate these values of which characterize the MTF. So we can characterize a detector property with this. Let's have a look at that, how this can be done. So until the final stage of the wave propagation, we are our result looks approximately like this. This is the case of the Zika virus, and after incorporating the noise, we can see that the projections look approximately like this. And afterwards, the MTF is applied, so the blurring of our detector. And if we have our noisy training data with known parameters of MTF, we can then use this scheme for optimization and find out the unknown parameters which I have been showing you, or the estimates of these unknown parameters, which I was showing you on the previous slide. Now here we can see that the loss, this is the focal frequency loss. So this is a comparison of images in the frequency domain, and with this focal frequency loss, that we have a parameter alpha, which we can use for tuning this loss function. And we have realized that with alpha equals approximately 0.5, we get the best results and are able to reconstruct the MTF function parameters. And this we can go further on the detector side, and as the detector is adding or incorporating the noise in our imaging process, we can also get or denoise the representation, so that we, instead of these representations, are able to obtain the noise-free ones in an optimal case. And again, the same methodology is used. We use an optimization framework, and through differentiability, we are able to then come up with a better and better representation of our specimen provided by the projections. Here we have one example of some tobacco virus, where you can see that we have a noisy projection as one example. Here we have a noise-free reference, so this is where we would like to get. And one way how you can actually get reduce the noise is simply to make many noisy projections and then average those, and by that you will be able to obtain an almost noise-free representation. In our case, we are using the optimization framework, and we can see that at least with the focal frequency loss, we are performing even a little bit better than the averaging. And in case the averaging is not usable, in case we do not know the ground truth, if we have real data, so that is out of question to be used, while in our case, we can still use our optimization-based approach in order to denoise with real-world data. I don't want to go too much into details. I will give you some resulting denoised representations of our tomographic reconstructions. So these were tomographic reconstructions from the denoised projections. And you can see here comparison of a noisy volume and performing direct volume rendering and the denoised volume, where you can see that a lot of noise has been already successfully removed from this acquisitions of SARS-CoV-2 variants. And this brings me to the last part. So we have been talking about the electron microscopy, tomography, and how we can create the volumes. And the volumes are eventually used, in our case, for 3D volume visualization. And if we do not perform a good reconstruction before, then the more noisy the visualization will be. But can we go even beyond that and try by means of deep learning methodologies to somehow interpret what is the noise and what is the specimen by providing enough of training data? And can we then incorporate this information into the volume visualization pipeline? That's the next part, which I would like to, or the last part, which I would like to cover. So here you can see that these are the slices of cryo-electron tomography, which have been created with the traditional reconstruction pipeline, but afterwards with some image enhancement. And you can see that after the image enhancement, by suppressing the noise and by basically binning, meaning that you are heavily making the resolution smaller and smaller, you can see that also will create us a little bit more denoised representation. And this is how, as far as we can get with the volume rendering. So we can see maybe some specimen there, but unfortunately there is still a lot of noise present. It's not very crisp. Now, our thought was the following. If we have provided that these interpretations of the individual elements in these scans can be interpreted and some segmentation can be done, in these deep learning approaches typically result in the so-called soft segmentation. So they give you a probability of how likely some structure, some voxel belongs to some class or some object in the volume. And in our case, that would be various elements of the SARS-CoV-2, such as the envelope or the internal part or the membrane-bound proteins like these spikes. And if we get these soft segmentation, can we use it for automatically defining the opacity transfer function for the rendering? That it was the research question we have been trying to answer with our research. So for the segmentation, well, in the most of the segmentation or interpretation is done on images in computer vision and computer graphics. In our case, that would be volumes, which is quite computationally intensive. But there are already some networks, architectures which support volumes and are useful for creating automated segmentation by means of inference of the learned network. So therefore, we first wanted to know whether such a network architecture, like for example in our case 3D-UNet, would be able to learn the various parts of the specimen, provided that we give it enough of training data, which we have generated and also validated with domain experts. The training data, which results, which are so-called pseudo labels, can be shown here. So we use a tool called Elastic, in which we define what is foreground, what is background, and what is the foreground structure. So it is the internal lumen, it is the envelope, or it is the red spike protein. And also with the red, yellow color, you can see the background. And then you specify this. It takes you for one volume, maybe something like half an hour, and then the Elastic will propagate these masks. You provide it for the entire volume for all slices, and this will be then further propagated within the volume, and this takes approximately something like 5 hours computationally. And we have done such a pseudo label generation for approximately 60 volumes of the SARS-CoV-2, which are coming from the same lab and with the same acquisition process. And you can see that these pseudo labels have been successfully generated. They look quite good for this particular volume. Unfortunately, if we try to propagate this information further to other previously annotated volume, this pseudo labeling approach fails, and we cannot use this the same approach for propagating the segmentation process on other novel volumes. So, but it was successful for completing the segmentation and creating pseudo labels, and then we use this interpretation for the training purpose. So you can see here how the pseudo labels of 4 classes looked like. So the 4 classes are background, spike, envelope, and the lumen, which is the internal part, the blue one. And you can see that the segmentations are, the pseudo labels are reasonably good. But for example, you can see that the pink is always surrounding this envelope. So somehow it leaks out, and the envelope is misclassified, partially being also the spike, which is not the case. You can see that artifact, for example here. Maybe I can also change them. So here you can see how the envelope is misclassified by this process. So the labels are good, but they are not optimal. And afterwards, but we can still see that if we use these pseudo labels as an opacity, then we will get rid of the noise. So if we think of just suppressing the noise, I think we have this already is a good indication that we will have quite noise-free representations if we would use these pseudo labels. So this was just application of pseudo labels, and we are just rendering here the mask of with somehow estimated threshold. We have an automatically defined transfer function specification and one threshold, which I will just tell you a little bit later. Now, if we are now taking these interpretations and use them for training the 3D unit, we will of course run into problem that our volume, which is 1024 1440 times approximately 200 to 500 slices along the Z, we will realize that we cannot really cope with this large data, and for that we have to use only smaller blocks of 128 cubed, which we are using, and we further subdivide our volume into so-called chunks, and these chunks are also overlapping. And within these chunks, we are then taking out these 128 powered by 3 blocks, and we use them for training. So for all the 60 volumes which we have. One epoch of this process, I think, takes approximately, of training, takes approximately 4 hours. And I think we are having running approximately 30 to 50 epochs. We are using the 3D unit network architecture, which consists of 18 layers, and it has this characteristic U shape. And it consists of the encoder part and the decoder part. And each of these parts are containing convolutional layers, ReLU, and also there are the skip connections. So you are having a connection between the different parts of the encoder and the decoder. For loss, we have been experimenting with various architectures. And by the way, this was not the only network architecture we have experimented with. We ended up with this architecture as being the best performing one and also being the fastest one for the training purpose. We used MSE loss, which also was the most providing the most satisfactory results. In total, we were using 60 volumes for the training, validation, and test. Afterwards, so this is an application of the segmentation on a novel set from these acquisitions provided by Tsinghua University. And we can see that the segmentations are actually better than the pseudo labels which we have seen before. This has also been confirmed by the domain experts who has mentioned that also the missing wedge problem. So I have to stop here. You would see that in some areas, and again, I need a red color for my pen. In some areas, like here, this would often be open, so there will be no envelope segmentation here or here, like it would look like here, for example. But with our deep learning approach, the inference actually results in much better handling the missing wedge problem. So this would be, therefore, these viral particles are often look like open here in these areas. That's actually the missing wedge artifact. But the deep learning can compensate for it and can close the particles quite successfully. Not in all cases, but in many cases, the result is significantly better than the pseudo labels and the results of segmentation with elastic software. Okay, so now we have our soft segmentations, meaning the probabilities of occurring of one or the other elements within our volume. But now we are integrating it into the volume visualization pipeline. So for each, now you can see a transfer function specification from previous research and utilization as somehow two things at once. Transfer function performs the interpretation, and also it then masks what do you want to see and what you do not want to see. So, and this part of the interpretation has been already carried out by the deep learning methodology in our pipeline. Now we only need to perform somehow the masking and estimate a very simple transfer function, just an opacity ramp, where we need to have some beginning of the ramp and the end of the ramp. Empirically, we have realized that end of the ramp is good to set to the probability 1. So if we are 100% confident about certain structure being that structure, then the opacity will be 1, and then on the range between 0 and 1 of the confidence, we find another value which will be the start of that ramp. And this another value is somehow a threshold. This value A is a threshold, and this will be the start of our ramp. And we identify automatically this threshold by an iterative optimal threshold estimation method, which in in a nutshell, works as follows. If we have an image, the assumption is that initially that the background is in the corners. So we take the values which are in the corners, average them, and that will be our representative value for the background. So so this will be, let's say, the iteration of BG1, and our corresponding A1 will be bigger as the value BG1, provided that the background is black or low intensity and the foreground is high intensity. Now, this is just heuristic to start with the edges, but the logic of this iterative thresholding approach is that we are looking for, so we generate with this threshold A1, we generate the particular image. And with that image, let's just imagine that we have like a noisy image of the letter A. And of course, this is now inverted because the A should be white and the background should be black. But we find the first iteration, and with the thresholding, there will be a lot of some noise, and it will look approximately like this. We will already be able to get some part of the background away, but there will be still a lot of background present in the noise, wrongly thresholded as being part of the foreground. Now, the gist of this idea is that we are looking at the background values which are close. So maybe I can magnify this here. So here we have a foreground structure after the first iteration, and we are interested in background values, intensity values around it. And that's what we are looking for the entire image in this case. And we are again creating so this will be the background estimation values 2. And we are again averaging them, and we will generate the threshold A2, which is bigger than the average of the BG2. So with this, we are more and more getting rid of the noise, and now we are at some point we are satisfied with the representation of our threshold, and this is how we estimate our threshold value for A for each of the slices, and then we are creating an average value of A so that we have one A parameter for the entire volume. So this is how we create the opacity transfer function, which is a very simple one, but it is simple because we already have done the tough part of interpretation into the soft labels segmentation. Now, the last part is the volume visualization. I will be quick here. So we are using traditional approaches for ambient occlusion. Imagine that you have some voxel and you would like to know how occluded is that voxel. So you are sampling the neighborhood of that voxel, and you are finding out whether some other voxels are very dense or are less dense, like this one, or are something in between. And based on this information of your neighborhood sampling, you will be defining how occluded your sample is. So this is the ambient occlusion of your samples in the volume during the direct volume rendering. And for the soft shadows, we are using something similar. So we are using both ambient occlusion and soft shadows for the illumination. And for soft shadows, we have a similar approach, but in our volume, we have a one voxel. And then if this is the light source, we are then, if this is the light source, then we can find out whether we are again sampling our neighborhood around this voxel, but we are sampling in the direction to the light source. So we are going towards the light source, and we are estimating how much of occlusion is coming from the neighboring voxels or samples, reconstructed samples, which are in the direction towards the light source. So that will give us an estimate on how much is this voxel in a shadow. And that results then afterwards in the following volume visualization, provided that we have done the inference into the 4 classes, and then we have performed the ambient occlusion or the soft shadows, and on top of that some post-processing filter. We are using the Bloom post-processing for further improving the visual appeal. We are defining these values of opacity transfer function automatically, but the user is free to change those and even turn off some of the channels if he or she likes to. Here you can see the details of the reconstruction of one such virion. You can see clearly the problem of the missing wedge artifact. So even if we have been able to close with our soft segmentation approach with 3D unit network the envelope quite well, we still have not completely closed it. So this is basically where we are at the moment. We have now the next step, the intermediate, very next step will be that this work on the 3D visualization of the Cryo-ET data has been done before our work on Cryo-ET tomographic reconstruction, both in the traditional algebraic reconstruction approaches with denoise, coupled with denoising, or with our differentiable microscopy methodology. So we have been using quite noisy projections. Now we have a lot of denoised data, and in terms of projections or the tomographic reconstruction. So also the learning process can be maybe more successful, even more successful, and maybe even the missing wedge artifact will be less pronounced. This is to be seen. So we have to use for our learning now these newly reconstructed volumes instead of the old tomographic reconstructions. And what is afterwards to come is that we have been showing that this differential microscopy simulator approach can estimate us the parameters of the detector, or perhaps can help us denoising the projection. However, if we will go even further, we can create and use this approach for creating the tomographic reconstruction. So that would be something we would like to try very soon in the future. And provided that that even works, we can have even higher ambitions. And these are maybe a little bit more far-fetched goals, like we can perhaps reconstruct an atomistic model with proteins, as you have seen in the beginning of my presentation, out of this cryo-electron tomography projections. And the next step, or the next step parallel to this goal, will be to create an end-to-end volume visualization pipeline, where the volume visualization will be done directly from the projections, and there will be no separate path for the denoising, for tomographic reconstruction, and then the inference. So to basically create a pipeline, and a learning-based pipeline, which does this all jointly, and at the input will be the Cryo-ET projections, and at the output will be vivid and noise-free 3D visualizations of that data. With this outlook, I would like to conclude my presentation, and I would like to thank my team, or our team at the NanoVis group at KAUST, in particular Gan, Feng, Julio, Cyril, and Onjay, And I would like to thank further other KAUST faculty and collaborators, and also international collaborators, without whom this research would not be possible. Thank you very much for your attention.
All right, thanks for Ivan's wonderful talk. Very professional technique for Cryo-EM volume data visualization. While the questions are coming from the audience, I will start for the first one. Ivan, I think it's very cool to combine reconstruction and denoise together. I think this will be a very exciting technology. But I have one question. I'm wondering if the early denoise will cause some important structure to be missed. You know, traditionally we first do reconstruction, then we do denoise. But what if you combine them together? Can you make some important structure to be missed?
So in fact, if you do the denoising, so imagine that you do the tomographic reconstruction first, you have a noisy volume, and then you perform denoising. You actually do not know how far you are to the original projections. You lose that connection. You only operate on your noisy volume, and you do not have the reference to the original projections. In our case, each iteration is basically using the projections to stay close to the ground truth. Now, you do not want to stay completely close to the ground truth of the projections, because those are noisy. So you are fine if you will have some error if you include this regularizer term. But this error needs to have a certain distribution within the volume. This error should, for example, be in some sense very uniform across the volume. Should not be very dependent on the specimen versus background location. Of course, it is a little bit, because the noise is directly dependent on the specimen, but there should not be a very strong variations of this noise. And if that's the case, or of this error term, and if that's the case, then you can be quite satisfied with the denoising approach.
I see. So you mean some kind of noise, our method might have some problem, but most of them, it works. Do you mean that?
Yeah. So, there is always a trade-off that you do not know whether the fine structures are noise or are actually fine structures of the specimen. That is not very clear. And in fact, one thing which could be useful in this context is to use the subtomogram averaging methodology. So by knowing that there are many similar structures in your data, you can actually use this concept of superimposition in this also denoising approach. And we have, I didn't mention it, but we have also been incorporating in this denoising the non-local means, which somehow act in a similar fashion, so that it takes into account some neighboring structure, and perhaps uses this superimposition principle in order to fight the noise and not the fine signal.
I see, I got it, thank you. Yes, so the second question is coming from audience. The people ask, can you explain a bit more about the pseudo labels? And could you using your first work, to simulate the label for training?
I think so. If we look at the pseudo labels, those have been manually created, and you can see several classes here. One class is the background, another class is the envelope, and another class is the membrane-bound protein, and all this is very laborious work. And also the processing, the computational processing, is also not negligible. So this is something which we would like to avoid and automatize if possible. And I think this is a good suggestion, that the background versus foreground segmentation creating soft labels out of this with using this denoising is actually a good idea. So that, but you cannot go further than that, because you know, discriminating then the internal part of the specimen and the envelope and the spike proteins, that will not be part of this denoising. But at least the two-class soft segmentation can be done with that approach. Yes, that's a good idea.
Thanks. I think there's another question from our audience in Zoom. Anyone have a question?
Ivan. Hi,
Okay.
I have a question. Hi, Ivan.
Hello.
This is Chong. This is Chong from Shandong University. Can you hear me?
Hello.
Hello.
I cannot see you.
Yeah,
I see.
yeah. My camera is yeah. Yeah,
That's
I don't
fine.
have camera now. So I have two questions. Yeah, I'm very interested in the third work, but now the segmentation, the rendering process, they are separate. It's not end to end, right? And
Yes.
so I'm thinking about, is it possible to improve the segmentation results by maybe using neural rendering or differentiable value rendering process? If yes, what do you think are the most challenging issues?
So you can see that maybe this differential microscopy simulation process can be seen as a neural rendering. And yes, if you have been, you know, if you are successful in reconstructing your structures, that can be used for the pseudo labels, even of the actual labels within your specimen, and then also the estimation of this of the transfer function is somehow explicit, and you know, we have some deterministic iterative approach which defines you this opacity estimation. But actually, we can also use learning-based approaches and train the visual mapping based on you know, training examples, training data. So we have not done that, and there are some initial works, or there are some works on that already. One I think even published at the last CSIG-VIS, which did differentiable volume rendering.
Yeah.
So that could be done. It has not been done end to end for our type of data, and we will be looking into that. This data has the problem that it's you know, the signal to noise ratio is very low. So generally, you cannot use techniques which work nicely on computed tomography. They will simply fail on this type of data sets.
So the ground truth labels for the segmentation are the pseudo labels. They're not assigned by users.
So we have done a comparison of the pseudo labels, which are, you know, semi-automatically generated. So you give them some hints as shown in this image, and then they populate the segmentations. And we have compared it with a ground truth segmentation, where we segmented an entire volume, and we have compared it, and it showed that the pseudo labels and the ground truth labels manual were very much similar to each other.
Yeah. I think it's very difficult to label segments in 3D space, especially when a signal and noise ratio is very low. Yeah,
but unfortunately, that is often done manually these days because of lack of better methods. We have been recently hosting people from like a tech support or user support from Thermo Fisher Scientific, who is the largest worldwide largest vendor of electron microscopy devices, and those have also now acquired the AMIRA software. So Thermo Fisher Scientific is developing the AMIRA software, which was originally developed at the SUSE Institute Berlin. So, and this is like a state-of-the-art of processing software for microscopy data and other data as well, but in particular microscopy. And they have concluded that with their tool set, currently, if you want to have a very good representations, you have to go fully ground truth, or you are satisfied with something what similar to approaches like our elastic. But I have the feeling that the elastic was performing even slightly better than the tool set which we experimented with in AMIRA.
Okay, great. Thank you for your great talk.
Thank you.
Okay, any more questions from audience? Any question from people in Zoom? Okay, if there's no question, I have a last question for Ivan. Yeah, I'm very excited for your work to combine differential rendering simulation with electronic Cryo-EM tomography together. This is really cool. I'm wondering, is it possible to find in the future put this kind of technique into hardware, into the tomographic hardware device? So then we can improve the quality, imaging quality, and while generating segmentation result automatically.
Okay, so you mean like, I mean, by hardware, I assume you do not necessarily mean that these algorithms have to be implemented on some FPGAs or something, but that these approaches are directly somehow interacting with the acquisition process itself, correct?
Yes.
So
yeah.
So you can, during the acquisition, you can change the acquisition so that you possibly get even a better result. That's an interesting thought, and we didn't go that far with our thoughts yet. But maybe that could be, it should be useful, I think.
Yeah. Maybe we can discuss that later. Yeah. Okay. Okay. That's our question we have now. Thanks for Ivan for your wonderful talk and answers. Thanks for our audience for joining us today. We will see you in next lecture on June 16 about the visual analytics for human-centered machine learning from Professor Andrew Enkel. Yeah. So see you in our next lecture. Thank you. Thanks, Ivan. Thanks for our audience. Have a good night.
Thank you very much, Yunhai, for having me today. Have a great day.
You too. Thank you. Bye. Yeah.
Bye.